Welcome back to microbiology, folks. Today, we're going to be covering Chapter 11 in 3 parts. And Chapter 11 is Characterizing and Classifying Prokaryotes, essentially a who's who within the prokaryotic world. So previously in this class, we've covered the history of microbiology, we've covered microbial genetics, nutrition, and growth. And that all allows us the terminology and the concepts to start to explore in more detail specific groups of prokaryotes and microorganisms and microbial agents and representatives of those groups. And so essentially what we'll be doing in Chapter 11 is taking prokaryotes and the domains archaea and bacteria, looking at them in general, and then into more specific groups, and then in representative genera of organisms. So I definitely recommend a note-taking technique that will allow you to start with broad concepts and then get more and more specific. In our next chapter, we're going to continue this theme by looking at microbial agents, viruses, and prions. We won't cover so much eukaryotic microorganisms in depth in this class, but I do invite you to look over Chapter 12 if you would like to look at that on the side. So we will also have an activity during this lecture in which we'll look at microbial reproduction, prokaryotic reproduction. It would be useful to have a piece of clay or dough nearby for that activity. So I'll invite you to pause the video, review previous content, preview Chapter 11 and the content that we're going to cover today, and gather any materials that you need, and then come back for the lecture. Okay, welcome back. So, as we know, prokaryotes dominate the world in terms of numbers of organisms. So there are about 5 times 10 to the 30th total prokaryotes on Earth. And even just on our body, there are upwards of 40 or more trillion organisms that live in and on us daily. Prokaryotes have co-evolved with Earth and other species on Earth since the beginnings of time, and they give us an idea of those interactions between species and between the environment, even over prehistoric time. And so to be able to look at prokaryotes is not just to understand prokaryotes themselves, but also to understand interactions with and between them. One of those interactions that we have with prokaryotes is pathology and disease, and so we will mention that throughout the chapter, but I do want to remind you that predominantly prokaryotes live amongst and between us in natural and healthy ways, and yes, there are implications of pathology, and pathology is important to, many of our kind of extremes interactions with them, and so we will mention that. But remember that they are existing around and in and on us naturally all the time outside of pathology as well. So as we talk about prokaryotes and classifying prokaryotes, we want to know a little bit about how prokaryotes are classified. So previous to this chapter, we've talked about nutrition and growth. And so some of those characteristics about how organisms are metabolizing, which sugars they metabolize, which fermentation or respiration processes they go through, those are all important characteristics in identifying how organisms are related to one another. Also, characteristics that we can see with our eye, like the shape and clustering of organisms, also really essential to categorizing prokaryotic organisms. Now, that is historically true, but in recent years, we've also developed some really powerful genetic technologies and tools that allow us to probe the DNA of organisms generally, and prokaryotes specifically as well. And so it is those tools that we have started to build more accurate phylogenetic categorization and classification based on. And so we've taken information about morphology, information about metabolism, and then we've added to it these really strong data around genetic information and phylogenetic relationships that we can see in DNA sequences. And so, for example, a clear macro example is In the macro world, we see a lot of similarities between birds and bats. And as we learn more about birds and bats, and we get genetic information about them, we know that they're very, very different in terms of their phylogenetic relationships. And those are two evolutionary kind of trails of flight that have emerged from very different groups. And that becomes really clear as we begin to interrogate birds and bats more fully. And so within the prokaryotic world, we see the same kind of distinctions emerging, where information that seemed clear in relation to morphology or visible characteristics that made us think that these organisms were related, once we get phylogenetic information about them and we probe them more carefully, we see that there's actually distinct phylogenetic differences between those groups. And so we'll run into that throughout the discussion today, and that gives you an idea of both the scientific method and the evolution of microbial technologies and techniques. Although we don't cover Chapter 8 specifically in this class, Chapter 8 does go over microbiological techniques and DNA recombinant technologies, and I would definitely recommend that you look over that chapter for more information on that. Okay. So what are some of the common morphologies of microorganisms? You've already interacted a little bit with these in lab and will continue to do so. But most commonly, we have cocci, which are spheres, or rod-shaped organisms, which are called bacilli, or spirillum, which are small, short spirals. But we also have variations among those shapes, where we have some organisms that kind of have a shape in between a coccus and a bacillus. We call those coccobacilli. We also have some variations of a rod-shaped organism where we have a curved rod, more like a comma shape, and those are called vibrio-shaped organisms. And we also have very long spirilla that are more flexible, and we call those spirochetes. Spirochetes have some very unique characteristics that are also related to their shape. We'll talk more about those today. You also have pleomorphic organisms, especially organisms that tend to have altered cell wall or cell membrane structures, and they have a little bit more freedom in how they present in the environment, and so they don't take on any of these kind of shapes specifically all the time. They can actually take on a number of different shapes depending on their environment or their growth, reproduction, so on and so forth. Speaking of reproduction, another really important characteristic is the arrangement of organisms, the arrangement of microorganisms within space. And so you have organisms that will be arranged in single file. You have organisms that will be arranged kind of on a flat plane in what we call tetrads, or you have organisms that are arranged in kind of random clusters. And it's not just that we're looking at these organisms and how they're kind of aggregating in a space or these beautiful formations that they're making. These forms and or arrangements or clusterings tell us about how those organisms reproduce. So if you have an organism that only divides along one single plane, like in this case here with this coccus-shaped organism, then they're going to be arranged in these single file chains. Whereas if you have organisms that are dividing on other planes, they will have other arrangements. So we're going to talk more about arrangement of cells in a minute, but I did want to preface that the type of division that we see is correlated with the arrangement that we see of these organisms within space under the microscope. Okay, and so we'll come back to this and we'll do an activity in a bit. But first, let's talk about reproductive strategies in prokaryotes. So in terms of reproductive strategies, as far as we know, all prokaryotes reproduce asexually. That means that the parent cell and the daughter cells are clones of one another consistently throughout archaea and bacteria. We did talk a little bit about horizontal gene transfer, where you have transfer of genetic information between peers horizontally, but we don't have sexual reproduction that we know of in prokaryotic organisms. Okay. The most common way that prokaryotes reproduce is through binary fission, where you have a parent cell replicate its genetic information, grow, and then allow that genetic information to move to the poles and then divide into daughter cells, and then continuing that process throughout. But we do have some other types of reproduction that tend to be characteristic to specific categories of microorganisms, and you'll see that as we move into our more specific discussions today. So, for example, snapping division, budding, spore formation, and motile filament clones. These motile filaments are really cool to watch. And so a little bit later on in the presentation, I will talk to you about cyanobacteria, and I'll recommend a short documentary called Pond Life. In that documentary, you can actually see videos of these motile filaments. Very cool. But first, let's go through these modes of reproduction. So here you have a video showing you binary fission, and it gives you an idea of how that process looks. And remember, this is the most common way that we see reproduction in prokaryotic cells. And in the lab, this is the most common way that we go from one colony forming unit on a plate or media, for example, to a whole colony that we can see visibly. This tends to be an exponential type of growth where we see lots and lots of clones that the organism can produce in a pretty short amount of time.
Bacterial cells do not divide by meiosis and mitosis. Instead, most bacteria grow asexually by a process known as binary fission. In this process, the bacterium copies its chromosome, elongates, and splits off into two daughter bacteria with a genetic makeup identical to the parental bacterium. Each daughter cell can continue the doubling process. This process happens rapidly, so the number of bacteria present in a solution can increase very quickly.
Okay. And here's a diagram that shows you that same process, where we have replication of the DNA, and then separation of the DNA to various poles, and then you have the separation of the daughter cells from one another, and then that can just continue on and on and on, where each daughter cell then goes through binary fission, and you can see how that would be that would lead to exponential growth within that population of clones. Okay. So we said binary fission is the most common method of reproduction in prokaryotes, but we have some other unique reproductive strategies that tend to be specific to various categories of organisms. For example, snapping division. So in this case, the parent cell, instead of kind of elongating a little bit and then dividing within the middle at a cleavage point, instead, the parent cell does replicate the genetic information and allow it to move to the poles, but the cell wall actually begins to form within the middle of that cell and just kind of forms and builds up and builds up and builds up, and that puts pressure on the original parental membranes and cell wall until they just snap, okay, to the top. And so usually just at whatever point is weakest, you see a separation of the two daughter cells, and then oftentimes they'll remain attached at one of the points along the cell wall. And so we'll get these very characteristic V shapes or side-by-side arrangement of either V's or kind of just parallel cells, which we call palisades. So V shapes or palisades under the microscope are telling us that this organism is probably reproducing through snapping division. Okay. We also have a process called budding, and budding is where the parental cell, it does replicate its genetic information, and then it makes an extension, which you can see here, of the original parent cell that has cytoplasm in it, and then it directs the replicated DNA into that extension, and then that extension kind of starts to grow as a bud and become larger and larger, and then separates from the parent, and you have this smaller cell that then is separated from the parent and then matures into a fully functional vegetative cell. Okay? And so we call that budding. Budding also occurs not just in prokaryotes, occurs in eukaryotes as well. So for example, yeast reproduce through budding also. Then you have a spore formation, which is unique to a group of organisms called actinomycetes. And essentially, you have an arrangement of chains or filaments of cells, and then those cells can actually make spores, multiple spores, that can then are kind of a dormant state, but then can become vegetative daughter cells whenever conditions are optimal. And so you can see filamentous active parental vegetative cells, and then these are lines of spores. Now, it's really important to distinguish these spores, which are true spores, from something that we're going to talk about in a little bit, which are endospores. So the distinction is that true spores are a reproductive strategy. And the way we know it's a reproductive strategy is that a parental cell becomes multiple daughter cells. Okay, so you can have any one of these parental filamentous vegetative cells producing a couple of or more than a couple of spores, and then those will then become vegetative cells. Whereas when we talk later about endospore formation, instead of it being a reproductive strategy, it's just a strategy for self-preservation. So one cell can make one endospore. When that cell dies, then that one endospore can become one vegetative cell. So really, instead of it being replication, where you get multiple daughter cells, it's just self-preservation. And that's a really important distinction because different categories of organisms will produce endospores from those organisms that will produce spores, true reproductive spores. Okay. So those types of reproduction are more common. There are a couple of really unique mechanisms for reproduction. For example, viviparity, which is live birth. So if you remember back to Epiloopicium, which is a relative of Clostridium, and this is an organism that, as we know, is the largest or some of the largest prokaryotic cells on Earth. They tend to actually exist in symbiosis with surgeonfish. And so here you see a very famous surgeonfish from the cartoon world. And so they, live in symbiosis with these fish predominantly, and they are very large cells that also are able to produce live young. And so remember, this is asexual reproduction. So the parent, as well as all the daughters, are all clones of one another. But the parent cell will reproduce multiple daughter cells within, I'm going to show you, within its cell. And then those cells, once the parent cell ruptures and dies, then those daughter cells will be released, and then we'll be able to grow into full-size vegetative cells. Okay. And so, let's italicize this. That's important. And so, as you see here, there are different mechanisms for reproduction in prokaryotes, and those mechanisms for reproduction, although binary fission tends to be the most common, you have some of these other unique modes of reproduction that tend to be telling as to who organisms are, because they tend to be unique to various categories of organisms. Okay, and so depending on the reproductive strategy, also depending on the plane by which those organisms divide, we can get specific arrangement of cells, which we had mentioned before, but we're going to talk a little bit more in a little bit more detail about now. Okay, so you have here a diagram that looks at cocci and a diagram that looks at bacilli. And essentially, if you have your organism doing binary fission, oh, here we go. So I just have a coccus-shaped piece of clay here, and I'm using a straight edge to then do binary fission just along one plane, right? Then you'll see that I'll get these single file adjacent cells that will kind of be produced there. Now I can just make these a little bit more round, a little bit more pretty there, so it's a little bit more obvious, right? So we start off with a diplococcus, as you can see here in the diagram. And then if each one of these continues to divide along that same plane, just one single plane that is consistent, then we'll continue to have this single file chain of adjacent cells, okay? And that tends to be unique to the organism. It'll divide on specific planes, or unique to the species, not to the organism. To the species, it'll divide on specific planes, and therefore it will have that specific arrangement. And we call this streptococcus. Strepto in reference to the arrangement where we have single file or cells one beside of the other, and coccus in reference to the circle shape of the organism. We could also have streptobacillus, okay? So in that case, we would have our cell, and the original parent cell, if it was a bacillus-shaped organism, then divides, again, just on one single plane. Okay, and then you get two adjacent cells that are daughter cells. That's a diplobacillus. And then if you continue that process, where each daughter cell just continues to reproduce by dividing on that same plane, then just like with our bacilli, with our cocci, we will have a single file chain, in this case of bacilli or rod-shaped organisms, and those will then be called streptobacilli. Okay, so you can see there our chain of bacillus-shaped organisms, and since it's a chain, strepto is in reference to the chain, and bacilli is in reference to the shape of those organisms. Okay, and so that you see that unique arrangement that's coming out of the reproductive strategy. Now, in this case, with B here in this image, instead of dividing again along one single plane, we have the organism dividing along two planes. And again, that's unique to the species or group of organisms. And in that case, instead of starting out with a diplococci, that then leads to a streptococci arrangement, we're actually going to start off with tetrads, so a group of 4 cells together. So for example, Micrococcus luteus, which we work with in the lab, produces tetrads, okay? You could have an organism that's dividing along 3 very distinct planes. These 2 types of division are usually unique to coccus-shaped organisms because they have that kind of consistent spherical shape. We don't see them quite as much in bacilli shaped organisms or rod-shaped organisms. So in the case where you had a coccus-shaped organism that's dividing along 3 clearly identified or clear planes, then you'll get duplicate tetrads, essentially, where we have 4 cells stacked in cuboidal arrangements or cube arrangements, and we call that sarcini, okay? You also have organisms that just divide along random planes throughout their coccus-shaped organism. And in that case, you'll just get this random clustering of cells, and we call that staph. So staphylococcus or staphylococci, where you have just random clustering of organisms, and that's all based on the planes on which they divide, okay? And then we talked about within bacillus-shaped organisms, we do sometimes have snapping division, where we have a rod-shaped organism, and then a cell wall is just kind of forms within the middle of the parent cell. At one point, it puts pressure on the parent cell and its cell wall, and then it just pops open. And then oftentimes they'll remain attached at one point, and we'll get these V-shaped cells under the microscope, or palisades, which is a side-by-side, either parallel or V-shaped arrangement of cells. Okay, and so I would recommend that you pause the video and then take a minute with your modeling clay or even dough that you have and go through and try to replicate these various arrangements of cells based on the planes on which those organisms are dividing. Okay. Okay, great. So feel free to add an image of your activity that you did as an optional addition to your discussion post for this class, for this chapter. Okay. So that's essentially a conclusion of our discussion on reproduction and arrangement. There is another really important characteristic that we want to talk about, we've mentioned already, that is important to classifying prokaryotic organisms and is unique to some groups of prokaryotes, and that is endospore formation. And so we talked previously about true spore formation as a reproductive strategy, whereas endospore formation is a unique mechanism of self-preservation in prokaryotic organisms, specifically bacteria within the Bacillus and Clostridium genus, genera, although there are some other genera that also produce endospores. And essentially, this is when the parent cell is in an unfavorable environment or condition and starts to feel like it might perish, right? And at that point, it will replicate its genetic information and encase it in an endospore that's very protective and very resistant to desiccation, very resistant to harsh environments, even in some cases resistant to UV and things like that. And that dormant endospore, it's not a vegetative cell, it's not an active metabolizing cell, it's just a dormant endospore. Kind of think of like a seed of a plant. When a plant produces a seed, and you do have many plants, when they're in harsh environments, they will produce more seeds, right? And then that seed is dormant until environments allow for it to regrow into a plant. So that's one way of thinking about endospores, right? Although it's not a reproductive mechanism like it is in plants. And so that one individual endospore that is within the parent cell will just remain in the parent cell until the parent cell dies. And then that endospore will exist on its own in the environment until conditions are favorable. And then that will, generate one vegetative cell that is metabolically active. And then it can then start to reproduce and metabolize and so on and so forth. Endospores are extremely relevant within the medical setting and also industrial settings because they're so resistant to microbial control, methods of microbial control. And then some of them also are from organisms that also produce toxins and so have some pretty extreme pathology. So endospore forming bacteria are the causal agents of diseases like anthrax, tetanus, gangrene, and some other really harsh kind of diseases. You can see here a parent cell with an endospore in the middle or at the end here. And so that gives us an idea of, maybe this organism is from these genera that tend to produce endospores. Okay. Also, just to note, the location of the endospore, whether it's central, subterminal, or terminal, is another indicator of who that organism is. So central endospores tend to be characteristic of the genus Bacillus, whereas subterminal endospores, which you can see here kind of more at the end of the cell, tend to be characteristic of Clostridium. Okay, and we'll talk more about both of those as we move through the discussion today. Okay. So that's our discussion generally of prokaryotes. Now what we're going to do is for the rest of part 1, we're going to talk about the domain archaea. So if you remember, prokaryotes are 2 separate, distinct, and diverse domains, archaea and bacteria. We'll finish our part 1 by discussing archaea. And then in part 2, we're going to discuss some representative bacteria. And in part 3, we'll continue that discussion. So just a reminder of the 3 domains of life: archaea, bacteria, and eukarya. Archaea and bacteria are prokaryotes that are simple unicellular organisms and oftentimes had looked very similar to one another under the microscope and even metabolically. And so up until 1977, so very, very, very recently, these were classified under one domain. And so in 1977, they were officially separated out as two unique domains of life with diversity, unique diversity within. And then eukarya is where we exist in kind of the branches of life where the cells are complex and compartmentalized, and we can have multicellular organisms within this category, although there are still unicellular eukaryotes as well. Okay. So let's look at this diagram of prokaryotic taxonomy. So you're looking at kind of how prokaryotes fall specifically in relation to their genetic diversity and their DNA and their phylogeny related to that. And also there's information on their metabolism and other characteristics that are kind of weaved within to those categories. And we can see based on genetic information that archaea is definitely unique. And then there are multiple different categories of bacteria that are unique and similar to one another. And so we'll start with our discussion of archaea, and then in part 2, we'll talk about deeply branching bacteria, excuse me, and gram-positive bacteria, and then we'll move into a discussion of gram-negative bacteria in the third part of this lecture. Okay, so we said that even like historically, archaea have kind of been confused with bacteria for a long time because under the microscope and even sometimes metabolically, they look similar to bacteria. But based on phylogenetic information, they are definitely unique. And even based on some of their characteristics, they do have a unique set of characteristics to archaea that are separate from bacteria. And so some of those are that their cell walls lack true peptidoglycan. They have structures similar to peptidoglycan, or even sometimes pseudopeptidoglycan, but they're not the same in terms of their chemical makeup as bacteria. And whereas all bacteria, well, almost all bacteria, except for a couple of genera that have lost their cell wall for various reasons, oftentimes because they're obligate parasites. But bacteria generally have cell walls made out of peptidoglycan, as opposed to this pseudopeptidoglycan or slightly different structure of cell wall in archaea. Also, the archaeal cell membrane has branched hydrocarbons within it rather than straight hydrocarbons, which is how bacteria's cell membranes are. And there are many archaeal cell membranes that are actually monolipid layers rather than bilipid layers. And so especially in extremophiles, that's really useful because it kind of maintains the integrity of the cell membrane within those environments. Also, in terms of codons in the genetic information, in archaea, across the board, AUG codes for methionine, whereas in bacteria, the codon AUG actually codes for N-formyl methionine instead. Interestingly, eukarya actually have an AUG codon that codes for methionine. So in this particular way, archaea are actually more similar than to eukarya than bacteria. Then there are 5 phyla of archaea, 2 predominant phyla, and then 3 that have fewer organisms within them. So Crenarchaeota and Euryarchaeota, predominant phyla of archaea. We have a number of different mechanisms for reproduction in archaea, including predominantly binary fission, but also budding and fragmentation. And you will see multiple different cell shapes from cocci, bacilli, spirals, and even pleomorphic organisms. Now here it says that archaea are not known to cause disease, and that's absolutely true. But also remember that we've just separated archaea as a domain as recently as 1977. And so we're still learning a lot about these organisms. We're still learning more about them. And it seems unlikely that there's in such a diverse group, there's absolutely no organisms that are pathogens of humans. although it is possible that the differences within their cell membrane and their structures and then their just evolution over time does prevent them from being pathogens. So this is kind of time will tell. There are a couple of organisms within our microbiome, there are archaea in our microbiome, like Methanobrevibacter smithii, which are implicated in some dysbiosis within the digestive system, for example. Not direct causes of pathology, but implicated in dysbiosis. So, like I said, time will tell. But as of now, as of what we know, there are no known archaea that are pathogens of humans. Archaea are a very diverse group of organisms. Some of those organisms are extremophiles, and now those organisms are some of the organisms that actually drew our attention more to archaea so that we could learn more about archaea. And so actually, historically, archaea have been thought of as an extreme group of organisms, extreme temperatures, extreme salinity, so on and so forth, although there's a significant amount of diversity. So there are organisms that are extreme, there are organisms that live in more moderate environments as well, that are all representative of archaea. And so we will talk about a couple of different groups of extremophiles. We'll also talk about methanogens in our discussion today. Okay. So you'll recognize here thermophiles, loving of extreme temperatures, usually extremely hot temperatures, and halophiles, loving of extreme salinities. And so we're going to talk a little bit about some of these organisms. So whenever you have an organism that lives in an extreme environment, it has to have the ability to maintain its integrity and homeostasis within those extreme environments, right? So if you remember our discussion of enzymes and also in previous classes, enzymes are really, really kind of temperature, pH, salinity specific. And when we perturb those things, then those proteins actually, and those enzymes start to unfold and they start to deactivate. Actually, a lot of what we do when we cook is manipulate the structure of proteins and enzymes. And once we manipulate them with temperature or acids, for example, then they actually become non-functional anymore. And so any organism that is able to withstand an extreme temperature, an extreme salinity, or an extreme pH has to have a way of stabilizing those enzymes or membranes or any macromolecules that are part of their structure, having the ability to stabilize those, okay? And so, like we said, with the monolayer in some archaea, of the membrane that allows them to have a stable membrane. Also, they'll have pigments that allow them to stabilize the various structures within. And so those organisms will then be able to survive and grow in those extreme environments. Another thing about extremophiles is that they tend to only survive within extreme environments because their anatomy is such that they can maintain their integrity at those extreme environments. If they are placed into a more moderate environment, their anatomy cannot withstand its integrity in those environments anymore, and they actually don't survive. So they only live in extreme environments. It's not that they can survive in extreme environments. They are only adapted to, over evolutionary time, those specific environments, okay? So we have Thermococcus and Pyrodictium, which you'll see in a minute, both organisms that exist in acidic hot springs, so actually extremes of temperature and pH, and that tend to be found in volcanic habitats or deep ocean rifts, for example, and they can withstand both high temperatures and acidic pHs. But the champion for thermophiles and actually being able to withstand highest temperatures is called Geogemma. Geogemma lives in deep sea thermal vents, and it's able to survive at a maximum temperature of about 130 degrees Celsius, which is about 266 degrees Fahrenheit, so it could essentially survive in your oven. But it actually grows and reproduces most optimally at a slightly lower temperature at about 120 degrees Celsius or 250 degrees Fahrenheit, which is still significant. So an organism like that could definitely withstand many types of microbial control, or an autoclave system or something like that, where we're trying to use heat to sterilize tools or media, so on and so forth, which is a concern. But the fact that they don't really survive at moderate temperatures anyways means that they're less likely to be an issue, because once those tools cool off, for example, or that medium returns to a moderate temperature, they're not likely to be able to survive and reproduce. But if there was an organism that could moderate that change in temperature and was pathogenic, that would become an issue. That would definitely be an issue. As of yet, we don't know of an organism like that. But such organisms are actually very useful to us within recombinant DNA technology and industrial settings. And that's because sometimes we need enzymes to function at higher temperatures. So many of you have probably done a polymerase chain reaction or PCR in prerequisite classes to this course. And if you remember, that is a procedure that allows for us to amplify a gene within an environmental sample, and that it happens within a thermocycler that just modulates the temperature from lower temperatures to higher temperature, lower temperature to higher temperature. And you have to have enzymes within that process that can actually withstand those higher temperatures and actually function at those higher temperatures. And so we isolate those enzymes from thermophilic archaea and bacteria, not just archaea, but archaea and bacteria. Same for, I don't know if you knew, but laundry detergents, and we'll talk more about this in the class as well, have enzymes in them to kind of eliminate some of the, lipids, for example, and there are lipases, so enzymes that break down fats or waxes that get on your clothes, for example. And so those need to be able to withstand higher temperatures within laundry cycles. And so we actually use enzymes off of these thermophiles for various purposes, and they're extremely important and relevant to that. So let's look at some of these organisms. Here we have Thermococcus. Thermo meaning withstanding high temperature, coccus, which is the shape of the organism. It also has these beautiful tufts of flagella that you can see there. Very beautiful. Then we have Pyrodictium, pyro meaning fire, so high temperature again. And Pyrodictium actually has these stalks that extend out from its body that allow it to actually hold on to sulfur granules, because it uses sulfur as its final electron acceptor. And so you can see this is more of a pleomorphic organism, so it has a different shape or different set of shapes, and those stalks that come out of it allow it to utilize sulfur in its environment. We had also mentioned that many of these extremophiles need pigments that actually protect them from those extreme environments. And from a macro scale, those pigments are beautiful. So you can see here in the Yellowstone National Park, archaeal mats and bacterial mats, probably a mix of both, but they have these pigments where you have this just beautiful coloration around this hot spring. Okay. Some other extremophiles are halophiles. And so halo meaning salt, phile meaning loving. So loving of extremely salty environments. And so just to give you an idea, human cells tend to be exist optimally within a salinity of 0.9% salt, 0.9%, okay? Whereas these organisms that are halophiles are existing within environments of above 9% salt, okay? And many halophiles will actually exist comfortably within a salinity of 17 to 23%. So very, very saline environments. And so we just know anecdotally that our cells can't withstand that. If we were to drink salt water, we'd actually dehydrate our cells. And so these are organisms that have a different osmotic balance where they can actually maintain their integrity within these very, very saline environments. And they actually would not survive a more osmotically neutral environment. Most of these tend to be classified within the phylum Euryarchaeota, and a lot of them have these beautiful red and orange pigments that allow them to withstand specifically protection from sunlight, but other extremes within their environment. The most well-studied of these that we know of is called Halobacterium salinarum, and it is really common in solar evaporation pools. So you can see in this picture from California, you have these solar evaporation pools, and they're just colonized by these archaea, so much so that you can see it from a satellite image. So very beautiful. Something that you'll notice from the name of the organism, we go back here, Halobacterium salinarum. Actually, this is salinarum. Halobacterium salinarum. So Halobacterium, because we had originally thought of these organisms as bacteria. And there tends to be, we try not to change names in the scientific world and in biology, because having a consistent scientific name for an organism allows us to consistently know what the research has been around that organism over time. And so, although there are some name changes that happen, sometimes you will see those relics within names that are maintained, so that we will have consistency between kind of the research studies that we have. Okay. Whenever there's a name change, there is the risk of actually losing some of the previous information about that organism, that, you know, its name has changed, and therefore, when you look at the different research about that organism, it's hard to know which is which. It can be confusing. So the final group of organisms that we'll talk about are methanogens. This is actually the largest group of archaea, predominantly in the phylum Euryarchaeota. There are actually a few organisms within this group that are actually thermophilic and halophilic as well. And these essentially are organisms that are able to convert carbon dioxide, hydrogen gas, and organic acids into methane. And they actually release methane into the environment. They exist within the environment in ponds, lakes, sediments, so on and so forth. But they also exist in ruminant animals, so your cows and deer and stuff like that. And particularly since we monoculture so many cattle for meat production and dairy, this can be an issue in relationship to climate change, because methane is about 25 times as potent as carbon dioxide is. And a cow's gut that has these methanogens in it can actually release, depending on what it eats, up to 400 liters of methane a day. And since we monoculture these in very, very large swaths, that's very relevant to climate change. So considering our connection to climate change via these archaea, these methanogens, is really interesting. And depending on how climate change impact is calculated based on cattle production, you have numbers between 15 and even up to 50% if you include both direct and indirect kind of mechanisms of contribution of methane, CO2, transportation, so on and so forth, deforestation that goes into cattle production. So if you're interested in more information around cow belching and the interaction with cows and archaea and climate change, there are a number of resources. One is We Are the Weather by Jonathan Foer. Jonathan Foer actually writes a number of books on cows and their relationship to climate change. So if you're interested, that's an outside resource for you. And that's kind of that final category that we want to talk about in terms of archaea. We've talked in this part about kind of an overview of prokaryotes, how we classify them, and then we've talked a little bit more specifically about some representative archaea. In the next sections, we'll start to talk about bacteria, and we'll first talk about deeply branching and gram-positive bacteria, and then in the third part of the lecture, we'll talk about gram-negatives. I would recommend that you kind of review the information on archaea using this Amoeba Sister video that's linked in the description below. And then I'll see you in part 2 of the lecture on chapter 11. Thanks for listening. See you in a bit.